So yesterday we were discussing about chloroplasts. We discussed the structure of chloroplasts. Now you know these chloroplasts, they are double membrane bound structures. In these chloroplasts, we have got those sac-like structures stacked upon each other. These sac-like structures which are stacked upon each other, they form the grana. And these sac-like structures, they are known as the thylakoids. There may be others as well. So these are the grana. This is a single granum. These grana, they are interconnected by membranes known as stroma lamellae or simply lamellae, or we also call them as frets, right? Now we have to look into this thylakoid. These sacs, they are known as thylakoids. So we have to look what is contained in a thylakoid. This thylakoid itself is a sac-like. This is a thylakoid. If we enlarge this thylakoid, it is a sac-like structure. On the inner surface of this thylakoid, there are small spherical structures. These small spherical structures which are present on the inner surface of the thylakoids. These small spherical structures present on the inner surface of the thylakoids, they are known as contosomes. They are known as contosomes. It is in these contosomes. This is the thylakoid. In thylakoid, we have got contosomes. It is in these contosomes that we have got the photosystem having a reaction center at its middle, at its center, and this is surrounded by chlorophyll molecules. There are chlorophyll molecules in it, right? This is in the center, reaction center, or we call it as photosystem. This is the reaction center, or we call it as photosystem. Photosystem, right? It is surrounded by chlorophyll molecules. Chlorophyll molecules, right? So this is a photosystem. This is a photosystem. These contosomes, this is a single contosome. It has got a photosystem or a reaction center at its center surrounded by chlorophyll molecules. In one contosome, there may be 230 chlorophyll molecules and 48 other carotenoid pigments. Carotenoid pigments. So where the center for photosynthesis is located, as I told you yesterday, this chloroplast, it is known as the kitchen of the cell. Why we call it as kitchen of the cell? Because it is here where the food for the plant is prepared, right? So that means photosynthesis is occurring. Photosynthesis does occur in a chloroplast. Where photosynthesis occurs in a chloroplast, the particular site for photosynthesis is a thylakoid. What is this thylakoid? It is a sac-like structure. On the inner surface of this sac, there are small spherical units known as contosomes. What is contained in a contosome? In a contosome, there is a reaction center or photosystem, right, which is surrounded by chlorophyll molecules. There are some approximately 230 chlorophyll molecules in a contosome, and there are some other pigments, 48 carotenoid pigments. Out of these 23 chlorophyll molecules, there are 160 chlorophyll A molecules, and there are 70 chlorophyll B molecules, right? So the total is 230. So in this contosome, there are 160 chlorophyll A molecules and there are 70 chlorophyll B molecules. So it is here where the photosynthesis occurs. So it is known as a photosystem or a reaction center. It is here where the quantas of light, they are trapped, and when the quantas of light are trapped, energy from them is utilized in combining this carbon dioxide and water. You know what is the reaction of this photosynthesis? In photosynthesis, we say it like this: 6 times carbon dioxide plus 12 times water. They combine in presence of sunlight and chlorophyll, right? And they form C6H12O6 plus 6 times oxygen. Oxygen is evolved as a byproduct plus 6 times water, right? This is the balanced reaction of photosynthesis, right? So here carbon dioxide and water, they are combined. Bonds are formed between carbon, hydrogen and oxygen by the help of energy which is trapped from sunlight. So where the sunlight energy is trapped, this energy of sunlight, it is trapped in this reaction center or photosystem by the help of chlorophyll molecules, right? And this trapped solar energy, it is utilized in formation of bonds between carbon, hydrogen and oxygen to form glucose, right? So the actual site for the formation of this glucose is thylakoid. So most of the glucose is synthesized in the thylakoids, right? So this is how these chloroplasts, they act as kitchens of the cell. So why we call them as kitchens of the cell? Because they trap the solar energy. By the help of that solar energy, they form bonds between carbon, hydrogen and water. When the bonds are formed, carbon, hydrogen and oxygen, they are combined and glucose is formed. So they form the food for the plant and for the other herbivores. That is why we call them as kitchens of the cell. So this is all about the chloroplast. I hope you have now understood what is the function of chloroplast. So the function of chloroplasts is to carry out photosynthesis. So they trap the solar energy. Where the solar energy is trapped? Solar energy is trapped in the thylakoids. What is contained in a thylakoid? In the thylakoid, on the inner surface of thylakoid, there are small spherical structures known as contosomes. In these contosomes are lying the reaction center or photosystem, where they are surrounded by chlorophyll molecules. So this contosome, it traps the contours of light. It traps the energy of sunlight. And that trapped energy of sunlight is utilized in the formation of bonds between carbon, hydrogen and oxygen to form glucose, right? So when carbon, hydrogen and oxygen, they are combined by the formation of bonds, they form glucose, and this glucose becomes as a source of food for the plant and for other herbivores, right? Now, can you tell me what is the function of chloroplast? So chloroplasts are the sites of photosynthesis. Number first, so they form the food for the plants and for the other herbivores. Number first function. Number second, these plants in the process of photosynthesis, they take up carbon dioxide from the atmosphere. So they reduce the levels of carbon dioxide in the atmosphere. If the levels of carbon dioxide increase in the atmosphere, you know that is going to cause global warming. So these plants, these green plants, they absorb carbon dioxide from atmosphere. They decrease the levels of carbon dioxide in the atmosphere, right? So they keep our atmosphere low in carbon dioxide content. And in the process of photosynthesis, they release oxygen as a byproduct. So we can say they take up carbon dioxide from the atmosphere and in turn they release oxygen into the atmosphere. So they add oxygen to the atmosphere, making the atmosphere neat and clean, making the atmosphere oxygen rich, which supports the life, and they consume the carbon dioxide of the atmosphere. So reducing the levels of carbon dioxide in atmosphere. Or we can say they maintain the balance of carbon dioxide and oxygen in our atmosphere, right? So number first, they form food for plants themselves and for the other herbivores. You know, plants are the producers. They provide food to all the other living organisms. If we remove plants from the planet Earth, then our planet cannot survive. Our planet cannot sustain, right? Because producers are the green plants. By photosynthesis, they manufacture food. They utilize this food for their own, and they also provide this food to other organisms. So this food is taken first by herbivores. From herbivores are taken by carnivores or consumers, right? So in this way, all the living organisms of the world, they get food by the process of this photosynthesis. So this is the most important organ. Number second, they maintain the ratio of carbon dioxide and oxygen in the atmosphere. If we plant more and more green trees, right? So those green trees will take up carbon dioxide from the atmosphere. They will reduce the levels of carbon dioxide in the atmosphere, and they will add oxygen in turn to the atmosphere, making our atmosphere neat and clean. So that is the function of chloroplasts. After chloroplasts, now we are going to discuss another cell organ. That is microtubules. Now another cell organelle, microtubule. These microtubules, they were discovered by Roberts and Franchi in 1953. Like ribosomes, these microtubules are again non-membrane bound organelles of the cell. They are also non-membrane bound organelles of the cell. We have discussed ribosomes. You know ribosomes, they are formed of only ribosomal RNA and proteins, and they are not covered by any membrane. So we call them as non-membrane bound organelles of the cell. Similarly, these microtubules, they are formed of a particular protein, tubulin protein, and they are also not covered by any membrane. So they fall in non-membrane bound organelles of the cell, right? Where do we find these microtubules in the cell? These microtubules, they form eukaryotic cellular components like cilia. So they are found in cilia, they are found in flagella, they are found in centrioles, they are found in sperm tail, sperm tail. They form spindle fibers, form spindle fibers. The spindle fibers which you observe during the process of cell division, those spindle fibers are actually microtubules. So they form spindle fibers, and they are also found free in the cytoplasm of the cell. In the cytoplasm of the cell. So where do we find microtubules? Microtubules can be found in some eukaryotic cellular components, eukaryotic cell organelles. The eukaryotic cell organelles like cilia, they contain microtubules in them. Flagella, they also contain microtubules in them. Centrioles, they also contain microtubules in them. Similarly, there are basal bodies. Basal bodies, they also contain microtubules in them, right? Then we find them in the sperm tail. The tail of mammalian sperm has got a bundle of microtubules in it that is known as exonium, right? So that exonium of sperm tail is formed of microtubules. At the time of cell division, we find that chromosomes are pulled towards the two opposite poles of the cell by the help of those fiber-like structures, which we call as spindle fibers. Those spindle fibers are actually bundles of microtubules, right? They are also found free in the cytoplasm of the cell, singly or in the form of bundles. Microtubules may be present in the cytoplasm of cell, either singly or in the form of bundles. They help the cell in the mobility, they help in the cell shape, etc. Right? They form the internal cytoskeleton of the cell, right? So these are the areas where these are the organelles where we find the microtubule. Now we will have a look of what material this microtubule is formed of and how do they look like, right? So we will try to see the structure of a microtubule. Let us see the structure of microtubule. So as the name indicates, they are small tube-like structures. That is why we call them as micro means small. Micro means small, and tubule means tube-like. So they are small tube-like structures, right? These small tube-like structures, they are formed of 13 protofilaments. They are formed of 13 protofilaments. Each protofilament is formed of tubulin dimers. Each protofilament is formed of tubulin dimers. This is alpha tubulin, this is another is beta tubulin. Alpha tubulin, beta tubulin. Alpha tubulin, beta tubulin. Alpha tubulin, beta tubulin. Right? So this is alpha tubulin. This is a beta tubulin. So these are basically protein molecules. These are protein molecules. Or you can call them as protein globules because they are globular in shape. So alpha tubulin and beta tubulin, they form a tubulin dimer. They form a tubulin dimer. This is one tubulin dimer. This is one tubulin dimer. So one tubulin dimer, another 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 tubulin dimer. So many tubulin dimers, they combine and they form a long thread-like structure. This is known as a protofilament. This whole structure, which is formed of repeating tubulin dimers, this is known as a protofilament. This is known as a protofilament. So this is one protofilament. Similarly, there is another protofilament. Similarly, there is another protofilament. There is another protofilament. There is another protofilament. How many such protofilaments combine? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and 13. So such 13 protofilaments, they combine and they form a small tube-like structure. This small tube-like structure becomes the microtubule. This becomes our microtubule. So this forms our microtubule. Are you getting me? So each microtubule is formed of 13 protofilaments. Each protofilament itself is formed of repeating protein globules known as tubulin dimers. Each tubulin dimer is formed of alpha tubulin and beta tubulin. So an alpha tubulin protein molecule, a beta tubulin protein molecule, these two tubulin protein molecules, they combine, they form a tubulin dimer. Then one tubulin dimer, another 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 tubulin dimer. Many tubulin dimers, they combine, they form one protofilament. One protofilament, another protofilament, another, 13 protofilaments. 13 protofilaments. These 13 protofilaments, they combine and they form a tube-like structure known as a microtubule. So we say that this microtubule is formed of 13 protofilaments. Each protofilament is formed of repeating tubulin dimers. Each tubulin dimer is having an alpha tubulin protein molecule and a beta tubulin protein globule, right? So alpha tubulin protein globule, beta tubulin protein globule, they combine, they form tubulin dimer. Tubulin dimers combine, they form protofilaments. Then 13 protofilaments combine, they form a microtubule, right? What is the length of this microtubule? How long it is? This microtubule may be anywhere between 0.2 to 25 microns in length. These microtubules, they are short. Microtubules are short. They may be 0.2 to 25 microns in length. What about the diameter of a microtubule? This microtubule is having a diameter of 25 nanometers. It is having a diameter of 25 nanometers, right? It has got an empty space in it. It has got an empty. Every tube has got a lumen. It also has got a lumen. This is the lumen of microtubule, right? This lumen of microtubule, it is 15 nanometers. It is 15 nanometers. Can you tell me then what is the diameter of this tubulin globule? Can you tell me then what will be the diameter of this tubulin globule? The diameter of microtubule is 25 nanometers. The diameter of its lumen, internal space, that is the 15 nanometers. Then what should be the diameter of this tubulin globule and this tubulin globule? Each tubulin globule has a diameter of 5 nanometers. So 5 plus 5, 10 plus 15, 25, right? So this is how microtubules are formed. This is how microtubules are formed. Now I hope you understand of what materials the microtubule is formed of. So these microtubules, they are formed of tubulin dimers, right? And these tubulin dimers, they combine, they form protofilaments. Then 13 protofilaments combine, they form a microtubule. A microtubule may be 0.2 to 25 microns long. It is having a diameter of 25 nanometers. It has got a lumen inside. This is the cap of my pen. This is its wall, and on its inner side, there is a cavity. This inner hollow cavity, this is known as the lumen. This is known as the lumen. So this microtubule is hollow internally. It has got a hollow cavity inside it. This is known as the lumen. Its lumen is 15 nanometers. And the thickness of each, the diameter of each tubulin globule, it is 5 nanometers. So 5 nanometer, 5 nanometer, 10 and 15, 25 nanometer, right? So this is the structure of a microtubule. Now what is their function? These microtubules, as I told you, they are found in cilia. So they form the skeleton of the cilia. They form the skeleton of cilia. You may have heard in cilia there are 9 plus 2 microtubules. The same is case in flagella. In flagella of eukaryotes, there are 9 plus 2 microtubules, right? So there are 9 peripheral microtubules and there are 2 central microtubules. So both cilia and flagella, they are formed of microtubules. Similarly, then we have got centriole. In centrioles, there are also microtubules. The microtubular arrangement of centrioles is 9 plus 0. In centrioles, there are 9 peripheral microtubules, right? In the center, they don't have any microtubules. So they are involved in the formation of cilia and flagella and centrioles. We are going to discuss about the structure of cilia, flagella and centrioles. So at that time, you will come to know where these microtubules are lying. They form the exoneme of sperm tail. You know, mammalian sperm has got a head, a neck, a middle piece, and a tail piece, right? So from the neck piece arises a bundle of microtubules, which runs through the middle piece and through the tail. That bundle of microtubules, which arises from the distal centriole, that is known as exoneme, right? So it is the sliding of microtubules in that exoneme, which makes the sperm tail to make movements, and by that movement of the tail, the sperm is able to swim, right? Sperm makes the locomotion, right? So this sperm tail, it also contains microtubules in its exoneme. Then we have got basal bodies. Those basal bodies are also formed of these microtubules. In meiosis and mitosis, we see that chromosomes, they are making movement towards the two opposite poles of the cell. What pulls the chromosomes towards the two opposite poles of the cell? As you might have seen many diagrams, there are small thread-like fibers which are attached to chromosomes, which are pulling the chromosomes towards the two opposite poles of the cell. Those small thread-like structures, which you might have seen in the diagrams during meiosis and mitosis, those small thread-like structures, they are the microtubules actually. We call them there as spindle fibers, but those spindle fibers, they are actually microtubules, right? So this is how microtubules are involved in formation of these structures. So that is all about microtubules. These microtubules, they have got a peculiar property. They can be easily formed and they can be easily broken. If tubulin dimers are added to these protofilaments, microtubules elongate. If tubulin dimers are removed from these protofilaments, suppose I remove these tubulin dimers, right? If tubulin dimers are removed from microtubules, then microtubules shorten. So they are not contractile. You should take it out of your mind that microtubules are not contractile. Microtubules can be elongated, they can be shortened. How they can elongate in length? By addition of tubulin diamonds to their protofilamentous. If tubulin diamonds are added to protofilamentous, microtubules elongate. If tubulin diamonds are removed from protofilamentous, microtubules shorten. So where these tubulin diamonds are added or where these tubulin diamonds are removed. For that, there are microtubule organizing centers near the centrosomes. There are MTOCs. There are MTOCs, microtubule organizing centers. Microtubule organizing centers. Where they are found? They are found near the centrosome. They are found near the centrosomes of the cell. So these microtubule organizing centers, they are present near the centrosomes of a cell. So here at the centrosome, I will show it in diagram. Let me show you it in diagram. Suppose this is an animal cell. And this animal cell is undergoing mitosis or meiosis. Okay, let it be mitosis. It has got chromosomes aligned at the equator of the cell. Let it be mitosis. So there are small thread-like structures. What are these small thread-like structures known as? These are the spindle fibers. As you know, there are two types: kinetophore spindle fibers and non-kinetophore spindle fibers. We will discuss it later. What are kinetophore spindle fibers and what are non-kinetophore spindle fibers? So these microtubules, they pull the chromosomes to two opposite poles of the cell. You know, in mitosis, the chromosome number remains same. Only the parent cell divides into two daughter cells, dividing the genetic material equally, right? So these microtubules, they are pulling the chromosomes towards the opposite poles of the cell. So how do they pull the chromosomes? These microtubules, they arise from somewhere around the centrioles. There are certain regions around the centrioles or centrosomes. These regions are known as microtubule organizing centers. There are some enzymes. Those enzymes, they either add tubulin dimers to the microtubules. When the tubulin dimers are added to the microtubules are formed, they are elongated. When tubulin dimers are removed from microtubules, these microtubules shorten. And it is the shortening of microtubules which pulls this chromosome towards this side, towards this pole. And on the other side, the chromosome is pulled on the opposite pole. So this pulling of chromosomes on two sides. This microtubule is pulling chromosome towards this side because the microtubules are shortening. Tubulin dimers are removed here at microtubule organizing centers, right? So this causes the shortening of microtubule, pulling the chromosome towards this pole. Here also the microtubules are removed, tubulin dimers are removed from the microtubules, shortening the microtubule, pulling the chromosome downwards, right? So when there is equal and opposite force, the chromosome breaks into two parts. The two sister chromatids, they separate. One sister chromatid goes to one pole and another sister chromatid goes to another chromosome, right? Another pole, right? So this is how they help in the process of meiosis and mitosis. They pull the chromosomes to two opposite poles by shortening of these microtubules. And microtubules can be easily shortened by removal of tubulin dimers, or they can be formed by addition of tubulin dimers. We are running out of time. This is section for today. Thank you.